imagine for a moment a detective. Let's just call him Medlock Holmes. Now, in the past, when Holmes was investigating the mysteries of the human brain, he would enter this, museum of structural imaging.
Right, he'd be looking at CT scans or structural MRIs.
Exactly. And in that museum, he basically examined the frozen architecture of the mind. It's exactly like looking at the blueprints of a grand, intricate building.
Yeah, you can see the walls, the corridors, where all the rooms are located.
Right. You can see if a wall has collapsed or, I don't know, if the ventricles are enlarged, but it is entirely silent. It is completely still.
Nothing is actually happening in those images.
Yeah, but today, you and I are following Medlock Holmes as he steps out of that static museum and enters a completely different space. We're calling it the Hall of Living Minds. And here, well, nothing is still.
It's a massive shift. I mean, the transition from structural to functional imaging was nothing short of a paradigm shift in psychiatry. We went from looking at the anatomy of the brain to literally watching the physiology of the mind in real time.
It's like walking into a room where the walls literally pulse with color as thoughts ignite across the cortex. You look over to the temporal lobe, and memory glows with this warm, sustained light.
Spot on. And in another corner, emotion flickers rapidly in the amygdala, or attention surges like a wave across the frontal networks.
Yeah. And language races through these intricate, overlapping neural pathways. Holmes is no longer looking at anatomy. He is revealing the dynamic, real-time conversations of the mind. And that is exactly what we are unpacking today for you. We're taking a deep dive into Chapter 12.8 of Kaplan and Sadok's comprehensive textbook of psychiatry, which focuses on functional brain imaging in schizophrenia.
And I think our anchor for this deep dive is a fundamental principle that you always have to keep in mind. Psychiatry is the study of how brain, mind, and experience interact, and crucially, how that process can go wrong.
Right. Structural imaging gave us the where, didn't it?
It did. It showed us the gray matter volume loss in schizophrenia or the enlarged ventricles. But functional neuroimaging, specifically functional magnetic resonance imaging or fMRI, well, that gives us the how.
It's really the ultimate translational tool.
Absolutely. It allows us to dissect these incredibly complex mental processes and bring cognitive and affective neuroscience directly into the clinical realm. We're essentially looking at how neural networks communicate.
How they miscommunicate, too.
Yes, and crucially, how they sometimes fall completely silent when a patient attempts to think or remember, feel, or even connect with someone.
Now, just to be clear for you listening, we aren't just, reciting facts on the textbook today. This deep dive is designed as a companion to the text, sort of a conceptual synthesis to build your mental models and strengthen your clinical reasoning.
Definitely read the chapter alongside this.
Yes, highly recommended. So let's start with the tools of the trade. Medlock Holmes needs his instruments to see these glowing networks, right? In the early days of functional imaging, the tool of choice was PT positron emission tomography. But Chapter 12.8 makes it very clear that fMRI is now the undisputed star of the show.
Without a doubt.
So walk us through that evolution. Why did we move away from PT, and what makes fMRI the preferred instrument in this Hall of Living Minds?
Well, the shift was largely driven by three things: safety, resolution, and practicality. I mean, PET scanning was revolutionary back in the 1980s.
It was a huge deal at the time.
Huge. For the first time, we could actually measure regional cerebral blood flow or glucose metabolism, but PT requires injecting radioactive isotopes.
Tracers, right, like fluorodeoxyglucose.
Exactly, FDG. You inject that into the patient's bloodstream, and then you have to wait for those tracers to reach a steady state in the brain before you can even take an image.
And obviously there's a limit to how much radiation someone can take.
That's the main issue. Because it relies on ionizing radiation, there is a very strict limit on how many times you can scan a single person. You just cannot ethically put a patient in a PET scanner repeatedly for longitudinal studies.
Or for those complex, multi-stage cognitive tasks, I'd imagine. Plus, the temporal resolution, the ability to see things happen second by second, has to be pretty poor if you're waiting for a radioactive tracer to reach a steady state over several minutes.
Precisely. PET gives you a beautiful snapshot of average metabolism over minutes, but, thought happens in milliseconds.
Right.
So fMRI, which came into prominence in the early 1990s, really changed the landscape because it is entirely non-invasive and involves absolutely zero ionizing radiation. It just uses magnetic fields and radio waves.
So you can scan someone as many times as you need to.
Exactly. Because of that safety profile, you can scan a person multiple times. Furthermore, fMRI offers much higher spatial resolution. We can localize activity down to just a few millimeters.
Wow, just millimeters.
Yeah, and vastly superior temporal resolution. We can track hemodynamic changes almost second by second. Plus, it's just more economical.
And doesn't it let you overlay the functional map onto the structural one?
Yes, directly onto a high-resolution anatomical MRI taken in the exact same machine during the exact same session.
It's brilliant. But let's ground this in clinical reality for a second. Because a functional MRI might be non-invasive mechanically, but it is intensely invasive psychologically. I mean, it is not a walk in the park.
Oh, not at all. I remember looking at some of the early fMRI data on schizophrenia from the late 90s, and researchers were constantly battling with motion artifacts and incomplete data sets.
Because the patients couldn't stay still.
Right. We really have to acknowledge the physical constraints of the technology.
Yeah.
The patient is placed on a narrow table and slid into a highly restrictive cylindrical magnet bore.
With the coil right over their face.
Yes, the space around their face is minimal. It can induce severe claustrophobia in anyone, let alone someone already struggling with psychiatric symptoms.
And the noise. I mean, the acoustic noise from the gradient coils in an MRI scanner can reach, 110 decibels?
Easily. It sounds like a jackhammer going off just inches from your head.
Right, so now imagine trying to perform a complex behavioral or cognitive task, like a working memory test where you have to hold a sequence of numbers in your mind, or an emotion identification task.
While lying perfectly still in a dark, noisy, confined tube.
Wearing bulky ear protection. I mean, for a healthy volunteer, it requires immense concentration. For a patient with schizophrenia, who might already be experiencing active paranoia or auditory hallucinations.
Or heightened anxiety, profound sensory processing deficit.
Exactly. It is an absolute monumental challenge.
Yeah.
So the subjective experience of the patient is a massive variable here.
It is, which means the data we get from these scans is incredibly hard-won.
Let's look closely at the actual signal we are fighting so hard to measure. The textbook highlights the bold-E signal blood oxygenation level-dependent imaging. This is basically the bedrock of fMRI, right?
It is the foundation, yes.
But the sources are very careful to point out that bold is an indirect marker. We are not measuring electrical action potentials. It relies on the magnetic susceptibility of deoxyhemoglobin. So how does this actually work at a biophysical level?
It is a beautifully elegant piece of physics, actually, discovered by C.G. Ogawa. So hemoglobin is the protein in red blood cells that carries oxygen.
We all learned that in basic biology.
Right. Now, when it is carrying oxygen, it is diamagnetic. That means it has no significant effect on the local magnetic field of the MRI scanner.
Okay, it's essentially invisible to the magnetic field.
Correct. However, when it drops off that oxygen to the tissue, it becomes deoxyhemoglobin. And deoxyhemoglobin has unpaired electrons, which makes it paramagnetic.
Meaning it disrupts the magnetic field.
Exactly. It creates these microscopic magnetic field inhomogeneities. These local disruptions cause the MRI signal from surrounding water protons to decay faster.
Oh, I see.
And that leads to a decrease in the MRI signal intensity on a T2 star weighted image. So more deoxyhemoglobin equals a darker image, less signal.
Okay, so let me trace the logic here. When a specific brain region, say, the prefrontal cortex suddenly becomes active because a person is performing a cognitive task, those firing neurons need energy. They consume glucose and oxygen.
Yes, they do.
So if they are consuming oxygen, the amount of deoxyhemoglobin should go up, right?
Right.
And the signal should go down. But that's not what happens in a functional MRI.
And that, right there, is the paradox that makes bold imaging possible. The body responds to the local neural activity by triggering neurovascular coupling. It dilates the local arterioles to rush oxygen-rich blood to that specific area.
But it doesn't just send what's needed.
No, the vascular system doesn't just deliver the exact amount of oxygen required. It dramatically overcompensates. The increase in regional blood flow vastly exceeds the actual regional oxygen consumption.
Okay, let's use an analogy here to lock this into our mental models.
Right.
We're essentially tracking a catering truck. The neurons are throwing a party, and they need food oxygen.
I like this.
But we can't see the partygoers. We can't see the neurons firing. What we can see is the delivery system. The brain calls for one pizza, but the vascular system sends like 10 catering trucks full of oxygenated blood to the site.
That is the perfect way to conceptualize it. Because there is a massive influx of 10 trucks of highly oxygenated blood, the relative concentration of the empty trucks, the deoxyhemoglobin, in that specific local area actually drops.
because the ratio changes.
Exactly. And since deoxyhemoglobin is what decreases the MRI signal, having a lower concentration of it means you get a net increase in the MRI signal intensity.
Mhm.
We essentially assume the party is happening wherever the catering trucks pile up.
We are looking for that tiny signal increase, which I think the text notes is usually just a 1% to 5% change in signal intensity, sometimes up to 25% in primary sensory cortices, but it develops over about 3 to 8 seconds after the task begins.
Right, that lag is called the hemodynamic response function.
But as brilliant as this Boldy proxy is, it has a major limitation, doesn't it? It only gives us a relative change. It tells us the signal went up compared to a baseline, but it doesn't tell us the absolute baseline blood flow.
That's a crucial point. That's why the chapter introduces a complementary technique, arterial spin labeling, or ASL.
How does that differ from Bold?
Well, BOLD is fantastic for mapping task-evoked activity, but because it relies on the ratio of oxygenated to deoxygenated blood, it is heavily influenced by venous outflow.
Oh, because the blood drains away.
Yeah, the signal often pools downstream in the draining veins, which can slightly blur the exact spatial localization of the neural activity. Arterial spin labeling takes a completely different approach. We use the MRI scanner's magnetic field to apply a radiofrequency pulse to the neck.
The neck? Why the neck?
We're magnetically tagging or labeling the water protons in the arterial blood supply in the carotid and vertebral arteries before they flow up into the brain.
Wait, so we put a magnetic barcode on the blood before it even enters the skull?
Exactly. We wait a brief moment for that tagged blood to perfuse into the brain tissue, and then we take an image. Then we take a second control image without the tagging.
And then you compare them.
Right. When we subtract the two images, the static background tissue cancels out entirely, and the difference that remains gives us a highly accurate quantitative measure of cerebral blood flow.
So ASL gives us absolute perfusion rates in genuine physiologic units.
Yeah.
Like milliliters of blood per 100 grams of tissue per minute.
Yes. It gives us a true baseline, which is incredibly useful for resting state studies in schizophrenia, where the baseline itself might be abnormal to begin with.
And it measures the blood flow directly in the brain parenchyma, the capillary bibs within the gray matter, rather than the downstream veins.
Exactly. It gives you a much truer spatial localization of the capillary perfusion. So Medlock Holmes has two magnificent magnifying glasses here. Boldy to look for dynamic, task-based changes, and ASL to measure the absolute baseline perfusion.
But having a magnifying glass isn't enough, is it? You need a strategy for how to interrogate the suspect. You need an experimental design. The source is heavily contrast block design with event-related design.
This is a vital distinction.
Let's break these down, because the choice of design fundamentally alters how we interpret the clinical data in schizophrenia.
If you want to map a neural circuit, you have to design the behavioral task very carefully. In the early days, almost all fMRI was done using a block design.
What does that look like in practice?
You present a single type of stimulus continuously for a sustained block of time, usually 20 to 30 seconds, and you integrate the bold D signal over that entire period.
So, for example, the patient is in the scanner, and for 30 seconds, they see a rapid succession of fearful faces on a screen.
Yes, and we might ask them to press a button every time they see a face. Then that active block is followed by a 30-second rest or control block, where they might just stare at a static crosshair on the screen or look at emotionally neutral shapes.
Just to give the brain a break.
Exactly. We then statistically compare the massive aggregate bold D signal from the active block to the control block. The advantage of a block design is its statistical power.
Because you're adding it all up.
Right. By piling all those stimuli together, you generate a very robust, loud hemodynamic response. It is fantastic for identifying the general circuitry involved in a broad domain, like the visual processing of faces.
But the textbook suggests there is a massive confounding problem with block design when we apply it to a psychiatric population, particularly schizophrenia.
Oh, it's a huge issue.
Because, think about it, if a patient with schizophrenia is taking a really difficult working memory test in a block design and they struggle with it, how do we interpret the scan? If their prefrontal cortex looks different from a healthy control, how do we know if their brain activity is fundamentally altered because of the neurobiology of the illness, or simply because they are failing the test, feeling incredibly anxious and basically giving up?
That is the Achilles heel of block design in clinical populations. You are capturing a 30-second chunk of data. If the patient is doing poorly, that block of brain data includes the neural signal of attempting the task, making an error, experiencing the emotional frustration of that error.
Feeling performance anxiety.
Exactly, and perhaps losing motivation entirely. You are muddying the waters. The hemodynamic signal of the illness is hopelessly entangled with the hemodynamic signal of failure.
That sounds like it was a crisis in early neuroimaging literature. We didn't know what we were actually measuring.
We really didn't, which is where event-related fMRI comes to the rescue.
How does that isolate the variables?
Instead of clumping stimuli into 30-second blocks, an event-related design presents individual stimuli sporadically and mixes them up. You might show a fearful face for 1 second, then a blank screen for a few seconds, then a happy face, then a neutral face, spaced out pseudo-randomly.
Okay.
We then use the scanner to time-lock the hemodynamic response to the exact moment a specific individual event occurred.
Because we know the hemodynamic response function takes about 3 to 8 seconds to peak, we can trace the signal back to that specific millisecond the image was shown.
Precisely. And the absolute superpower of this design is that we can look at the behavioral data, we can see which button the patient pressed, and we can retroactively separate the neural activation associated with correct responses from the activation associated with errors.
Oh, wow. That is brilliant. So we can just filter out the noise of failure. We can look solely at the moments the patient successfully identified the fearful face.
We level the playing field regarding performance. We compare the brain activity of a healthy control getting the answer right with the brain activity of a patient with schizophrenia getting the answer right.
So you control for the nuisance variables of frustration, confusion, and error monitoring.
Right. And if we still see differences in the brain circuitry during successful trials, which, spoiler alert, we do in schizophrenia, we can be vastly more confident that we are observing the underlying neurobiological trait of the illness rather than just the state artifact of poor performance.
It allows us to isolate the trait from the state. That is a vital piece of clinical reasoning. But even as our experimental designs evolved, the way we analyze the massive data sets generated by fMRI underwent an even bigger revolution, didn't it?
It really did.
The textbook outlines a paradigm shift from what neuroscientists jokingly call blobology to network connectivity analysis and graph theory.
Ah, blobology. That was the era of localization. We used to look at fMRI scans the same way we looked at structural lesions in neurology. We would ask which specific area of the brain didn't light up.
You're just looking for isolated blobs of color on the statistical map.
Exactly. We thought if we could just find the precise coordinates of the schizophrenia lesion, we would solve the puzzle.
But the brain isn't a collection of isolated islands. It doesn't work like a phrenology map where one specific lump handles memory and another lump handles fear.
No, not at all. The brain operates as highly integrated distributed networks. The true revolution in functional imaging came when we stopped asking, where's the lesion? And started asking which conversations have broken down.
I love that framing.
We moved to functional connectivity analyses. This involves looking at the low-frequency temporal correlations in the Boldy signal between different brain regions. If region A in the frontal lobe and region B in the parietal lobe are fluctuating in their oxygen demands in perfect synchrony over time, we infer that they are functionally connected.
They are talking to each other.
Exactly. And graph theory takes this conceptual shift and gives it mathematical rigor.
I want to spend some time here because graph theory sounds incredibly intimidating. It sounds like high-level computer science, but it's actually a very intuitive way to map complex systems, whether it's the internet, the flight routes of an airline, or the human brain. How does graph theory apply to fMRI?
At its core, graph theory reduces complex systems to two simple elements, nodes and edges. A node, or a vertex, is a distinct point in the network. An edge is the connection between two nodes.
So in neuroimaging, a node is a specific brain region, say a cubic centimeter of tissue in the anterior cingulate cortex.
Yes, and an edge is the statistical strength of the functional connectivity between that node and another node.
So if the temporal correlation of the Boldy signal between two nodes is very high, that's considered a very strong edge.
Yes, and by mapping out thousands of nodes and millions of edges, we can apply mathematical algorithms to understand the architecture of the brain. We look for hubs highly connected nodes that act as central routing stations, much like a major international airport.
And what's small world architecture? The text mentions that.
Small world architecture is a network design that balances local, specialized clusters of nodes with long-distance edges that allow for rapid global communication across the whole brain. It's incredibly efficient.
And when we apply this graph theoretical maths to the resting brain, when a person is just lying in the scanner not doing any specific task, we discover macro-level networks that fundamentally organize human consciousness.
We do. We identify the default mode network, or DMN, anchored in the medial prefrontal cortex and posterior cingulate. That network is highly active when we are awake but internally focused, introspecting, or daydreaming.
Okay, so that's the internal network.
Right. Then we find the central executive network, the CEN, anchored in the dorsolateral prefrontal cortex, which handles demanding external cognitive tasks.
The external network.
And crucially, we find the salience network, involving the anterior insula and anterior cingulate. That essentially acts as the switchboard operator, detecting important stimuli in the environment and switching the brain's resources between the internal default mode and the external executive network.
That's a beautiful system. And as we will see in schizophrenia, the nodes themselves, the individual brain regions, might be structurally completely intact, but the edges are frayed. The network topology is altered.
The routing is off.
It's like having a perfectly functional telephone in your house, but the regional exchange is completely misrouting your calls.
That's a great way to put it.
But, before we explore those specific broken conversations, we need to acknowledge the noise in the signal. The textbook outlines several clinical variables that make fMRI research in schizophrenia incredibly difficult to interpret. What are the major confounds?
Well, the primary hurdle is just the immense clinical heterogeneity of the disorder itself. Schizophrenia is almost certainly not a single monolithic disease entity. It is a clinical syndrome with very diverse etiologies. When you put 40 patients in a scanner, their symptom profiles vary wildly.
Right. Some might have severe positive symptoms like auditory hallucinations, while others are dominated by negative symptoms like avolition and allogio.
And their demographics matter significantly. Gender is a powerful moderating variable. Men typically experience an earlier onset of the illness. They tend to have more prominent negative symptoms and often show different patterns of structural and functional brain abnormalities compared to women.
Age and chronicity of illness must be massive factors too.
Oh, definitely. A brain that has experienced 20 years of chronic psychosis looks very different functionally from a brain in its very first episode. If your research sample isn't meticulously balanced and matched to controls, your data will just skew.
And then there is the elephant in the MRI suite. Antipsychotic medications. Nearly every patient participating in these studies is on medication that fundamentally alters brain chemistry.
It is an unavoidable confound. The vast majority of our functional imaging data comes from medicated patients. Antipsychotics act primarily by blocking dopamine D2 receptors, but many also affect serotonin, histamine, and adrenergic receptors.
And those neurotransmitter systems intimately regulate cerebral blood flow.
Exactly. Therefore, the drugs themselves directly alter the bold B signal. Unless you are studying first-episode neuroleptic naive patients, which is logistically and ethically very difficult to do in large numbers, it is a constant, painstaking statistical challenge to disentangle the neurobiology of the illness from the iatrogenic effects of the treatment.
We also have the Goldilocks problem of task difficulty, which we touched on a bit with block designs.
Right. If you make a cognitive task too hard for patients with schizophrenia, they fail, and you introduce the noise of anxiety and off-task brain networks lighting up.
But if you try to control for performance by titrating the task down, making it incredibly simple so that patients perform just as perfectly as healthy controls, you run the risk of fundamentally changing the cognitive process you are actually trying to test.
That's exactly the problem. If you make a working memory task too easy, it might no longer recruit the dorsolateral prefrontal cortex at all. The patient might just be using basic visual recognition.
So you solve the performance confound, but you've lost the neural circuit you were hunting for. It's a delicate, maddening balancing act for researchers.
But despite all of these methodological challenges, the noise, the claustrophobia, the medications, the field has mapped out some remarkably consistent and profound findings.
Which brings us to the core of the clinical data. Let's dive into clinical manifestations, starting with cognition. Our sources highlight that schizophrenia features diffuse, widespread cognitive deficits, with particularly devastating impacts on executive function, working memory, and verbal episodic memory.
Yes.
So Medlock Holmes is looking at the functional scan of a patient with schizophrenia who is attempting to learn and encode a list of words. What does he see in the networks?
He sees a very specific pattern of disrupted communication. During the learning or encoding phase of verbal memory, patients with schizophrenia consistently show decreased activation of the frontal cortex, specifically the inferior prefrontal regions. This was one of the earliest and most robust findings. We call it hypofrontality.
But remember, We're thinking in networks now. The frontal lobe doesn't act alone.
Exactly. The core finding isn't just a quiet frontal lobe. It's a dysfunctional handshake between the front of the brain and the temporal lobes.
The frontotemporal system. This is crucial. Walk us through the mechanics of this handshake in a healthy brain versus a brain with schizophrenia.
Well, in a healthy neurotypical brain, when you are performing a demanding verbal memory task, your prefrontal regions heavily activate. They need to orchestrate the strategy, organize the information, and apply semantic labels.
Right.
Simultaneously, your superior temporal gyrus, which is heavily involved in basic auditory processing and language, actually deactivates.
Really? It turns down.
Yes, it is a highly coordinated push and pull. The prefrontal cortex essentially tells the superior temporal gyrus to quiet down so it can concentrate on encoding the memory into the hippocampus.
Filtering out the noise. But in schizophrenia, that suppression fails, doesn't it? The superior temporal gyrus doesn't deactivate.
No, it doesn't.
It's like the temporal lobe is refusing to quiet down, leaving the prefrontal cortex struggling to do its job amidst the static.
Precisely. And functional connectivity analysis, those time series correlation studies conducted during verbal and coding tasks, reveal exactly where this communication breaks down. Researchers found that patients with schizophrenia had significantly reduced functional connectivity between the dorsolateral prefrontal cortex, the DLPFC, and temporal lobe areas like the parahippocampus and the superior temporal gyrus.
Let's anchor this with our telephone analogy. The phone lines are cut. If the DLPFC is the high-level manager of the company, trying to organize and strategize the incoming information, and the temporal lobe is the worker on the factory floor who actually receives and stores the memory, their direct, secure phone line is severed.
Yes.
The temporal lobe is generating noise, and the manager can't reach them to give instructions.
That captures the functional consequence perfectly. The primary, most efficient communication channel is severely attenuated. But the brain is highly adaptive. It doesn't just give up. It attempts to compensate.
What does it do?
The same connectivity study showed that while the DLPFC temporal connection was reduced, there was a simultaneous increased functional connectivity between a different area, the ventrolateral prefrontal cortex, or VLPFC, and those exact same temporal regions.
Okay, wait, so the factory worker's phone line to the main manager is cut. So the worker tries to call a different manager, the VLPFC. They dial a backup number to try and get instructions.
That's what it looks like on the scan.
That sounds like a brilliant adaptation.
Yeah.
Does it work?
And that is the absolute tragedy of this broken conversation. No, it does not work. In healthy participants, strong connectivity between the temporal lobe and the DLPFC strongly correlates with better memory recognition later on. It is an effective circuit. But in patients with schizophrenia, this increased compensatory connectivity with the VLPFC does not correlate with better accuracy or memory performance.
It is an entirely ineffective compensatory effort.
They are dialing a backup manager who simply doesn't have the appropriate authority, the right algorithms, or the proper cognitive tools to organize the memory properly. The metabolic effort is there. The network is actively trying to rewire itself on the fly, but the functional, behavioral outcome remains severely impaired. The memory is not encoded efficiently, and the patient fails the recall test.
That is a staggering insight into the pathophysiology. It's not just a void. It's not just a lack of activity. It is an active, desperate misrouting of neural resources that ultimately fails.
It really is.
And this issue of misrouting and failing to suppress noise isn't just about high-level complex memory tasks. The textbook describes issues much earlier in the information processing cascade, right down to early sensory integration.
Yes, and this is beautifully mechanically demonstrated by what we call oddball paradigms.
How does an oddball task work?
An oddball task is a classic neurophysiologic probe. We put the patient in the scanner and present a continuous sequence of standard, boring, repetitive stimuli, like a series of identical tones or identical blue squares on a screen.
The brain quickly habituates to these, right? It stops paying attention.
Exactly. But randomly, you insert a target oddball, say a red square, that the patient has been explicitly instructed to detect and respond to by pressing a button. And occasionally you insert a novel, completely irrelevant, highly distracting oddball, like a sudden flash of a checkerboard pattern that they are supposed to ignore completely.
So if we're doing auditory tones, beep, 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 B-O-B, that's the target you need to catch. Beep, 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 P-arc, that's the novel distractor you need to ignore. What do the fMRI scans show when patients with schizophrenia attempt this?
We see a tale of two distinct deficits that perfectly mirror the clinical phenomenology of the illness. For the target detection, the BOP patients with schizophrenia showed diffuse cortical hypofunction.
They don't react enough.
Right. They have an attenuated, weakened hemodynamic response across the prefrontal and parietal regions that are supposed to light up.
We interpret this as insufficient top-down activation. The brain's executive networks, the central executive network we talked about, aren't adequately priming the sensory systems to look for the target. They aren't turning the spotlight of attention on.
Exactly. They miss the cue. But the reaction to the distractor is where it gets truly fascinating. When presented with the novel irrelevant distractors, the bark, the patients do the exact opposite.
They overreact?
They overactivate distributed sensory and frontal areas, particularly regions related to visual spatial processing and working memory.
They underreact to what is clinically important and explicitly instructed, and they completely overreact to what is irrelevant and supposed to be ignored.
Yes. The clinical meaning here is profound. They suffer from an attenuated response to targets combined with an overprocessing of bottom-up irrelevant events. Their brain is struggling to filter out the noise of the environment. The gating mechanism is broken.
If we extrapolate this from a scanner into the real world, it explains so much of the subjective lived experience of schizophrenia. The feeling of being constantly overwhelmed by sensory input. The inability to focus on a conversation in a crowded room.
It's exhausting for them.
And crucially, the delusional salience assigned to random events. I mean, if your brain is overactivating its memory and spatial networks every time a car backfires or a stranger wears a bright yellow shirt, you're going to start thinking those irrelevant events have deep personal meaning.
You construct delusions to make sense of the aberrant neurobiological salience your brain is generating. You can see how this simple oddball fMRI data maps directly onto the complex psychiatric presentation. They cannot gate the sensory flood, and the top-down cognitive control is too weak to correct the errors.
This brings us to a major transition in our conceptual synthesis. We've looked at cognition, where the phone lines are cut and the sensory gates are broken. Now, if the brain is this bad at routing basic working memory and auditory tones, what happens when it has to process something vastly more complex, ambiguous, and potentially dangerous like human emotion?
Oh, this is a fascinating area.
This is Clinical Manifestations Part 2, Emotion and Social Cognition. And Medlock Holmes has discovered a massive paradox here. Let's call it the irony of the amygdala.
This is perhaps one of the most counterintuitive, revealing, and frequently misunderstood findings in all of psychiatric neuroimaging.
Clinically, a hallmark negative symptom of schizophrenia is flat affect, or blunted affect. It is a severe diminution of emotional expression. The patient's face might resemble a mask. Their voice might be entirely monotone, lacking prosody or inflection. They appear completely disconnected from the emotional weight of a situation.
That's the classic clinical presentation, yes.
So, logically, intuitively, you would expect their emotional center, the amygdala and the broader limbic system, to be dormant, scarred, or underactive.
Right.
But when we put them in the fMRI, the data shows something entirely different.
It does, and the history of how we discovered this hinges entirely on the experimental designs we discussed earlier. If you use a traditional block design, where you give them a top-down explicit task like push a button when you see a sad face, and you aggregate the boldy data over 30 seconds, you actually do see diminished limbic activation overall compared to healthy controls. The circuit seems quiet.
Which confirms the clinical bias.
But when researchers refined their tools and used an event-related design to look at the specific, immediate, time-locked reaction to threat-related stimuli, specifically angry or fearful faces, they uncovered the paradox.
What did they find?
They found that patients with schizophrenia exhibited abnormally increased, exaggerated bottom-up amygdala activation in those first few milliseconds.
Hold on. Their amygdala is firing more intensely to a scary face than a healthy person's amygdala.
Yes. It is hyper-responsive. And here's the clinical mind-bender that completely flips our understanding of flat affect. In a healthy, neurotypical individual, a higher spike in amygdala activation during an emotion recognition task portends a correct identification of fear. Your internal alarm goes off and you correctly say, that person is terrified.
Makes sense.
But in patient with schizophrenia, higher amygdala activation correlates with an incorrect response. They misidentify the emotion.
Let me push back on this because it feels completely backwards. The stronger their emotional center reacts to a scared face, the more likely they are to get the answer wrong. And as the textbook notes, this hyperactive amygdala response is strongly correlated with more severe clinical ratings for flat affect. Why? How does a hyperactive emotion center cause a blank face and an incorrect answer?
It feels backwards until we apply advanced connectivity analysis, specifically structural equation modeling, to look at the directional flow of information. Structural equation modeling allows us to test mathematical hypotheses about which node is driving the activity of another node.
Okay, so what is it showing us here?
What we are seeing in schizophrenia is essentially a catastrophic short circuit caused by an overloaded network.
Let's break that down mechanically.
Let's use our alarm system analogy. The amygdala provides a rapid, primitive, bottom-up alarm signal that a threat is present in the visual field. This signal is fast and dirty. That signal then has to travel upwards to the top-down cortical regions, the prefrontal and anterior cingulate cortices, which evaluate the threat in context, categorize it linguistically, and formulate an appropriate cognitive and motor response.
So the amygdala screams danger,
Mhm.
and the prefrontal cortex looks at the context and says, calm down, that's just a photograph of an angry face on a computer screen. We are safe inside an MRI scanner. The emotion is anger.
Precisely. The cortex contextualizes and regulates the amygdala, but structural equation modeling shows that in schizophrenia, that bottom-up amygdala connectivity is massively excessive. The alarm isn't just loud, it's deafening.
Oh, wow.
It is a torrential flood of neurochemical signaling. It is so loud that it disrupts and completely short-circuits the coordinated activation of the cortical regions required to process it.
The prefrontal cortex is blown out by the surge.
The top-down categorization fails because it is overwhelmed by the bottom-up interference. The patient gets the multiple-choice answer wrong because their cortex literally cannot process the massive, chaotic surge of amygdala activation. They cannot apply the semantic label of fear because the cognitive circuits are jammed by the emotional static.
And how does that explain flat effect?
Flat effect can be fundamentally re-understood as a downstream consequence of this constant, overwhelming internal alarm. The system shuts down externally because it is overloaded internally. It is a protective, compensatory withdrawal.
That is profound.
The face goes blank and the voice goes flat, not because they feel nothing, but because the internal limbic response is so chaotic, terrifying, and disruptive that it paralyzes the outward expression pathways.
That reframes the negative symptoms of schizophrenia entirely. It's not an empty room. It's a room where the stereo is turned up so loud the speakers have blown out and the amplifier has gone into protective mode.
It is a profound shift in clinical empathy, I think. Furthermore, because the amygdala is intimately linked structurally and functionally with the hippocampus, this excessive, unregulated emotional arousal likely disrupts episodic memory formation.
Oh, connecting it back to cognition.
Yes. This provides a unified mechanism that explains why emotional dysregulation contributes to the cognitive memory deficits we discussed earlier. The noise from the amygdala corrupts the hippocampal encoding process.
This is a brilliant deconstruction of the phenotype. So if the amygdala is hyperfiring, what triggers it? The sources suggest there are specific visual modulators we can look at, particularly regarding how faces are processed at a sensory level.
Researchers are actively trying to construct mechanistic models of this fear response. To understand it, we have to look at the visual pathways. The visual system processes information in two main streams: the parvocellular pathway, which handles fine detail and high spatial frequency, and the magnocellular pathway, which handles broad strokes of light and dark, motion, and low spatial frequency.
The magnocellular pathway is the fast, blurry system, right?
Yes. And fMRI studies have shown that in healthy brains, the amygdala response is exquisitely tuned and potentiated by low spatial frequency information, the blurry, broad strokes, specifically in the upper part of the face, the eye region. The amygdala is biologically hardwired to look for the whites of the eyes to detect fear.
Which makes evolutionary sense. You need to know instantly if someone is looking at a predator without waiting to process the fine details of their eyelashes. Gaze direction matters too, right?
Absolutely. Directed fear, someone looking directly at you with a terrified expression, or averted anger, someone looking away from you in anger, are highly ambiguous social threats. Am I the threat? Is there a threat over there? These ambiguous gaze directions spike amygdala activity in healthy people because they require more processing to decipher.
Now, bring this back to our patients. How does this visual processing fail in schizophrenia?
Well, we know from classical neurology that patients with bilateral amygdala lesions like in Urbach-Wiethe disease naturally avoid looking at the eyes of faces. They look at the nose or the mouth. We also know from eye tracking studies that patients with schizophrenia demonstrate poor eye contact and actively avoid scanning the eye region when looking at faces. Since fear identification relies almost entirely on cues from the eyes, researchers hypothesize that the emotion recognition deficit in schizophrenia might stem from a fundamental impairment in attending to or processing changes in the eye region.
They're looking at the mouth, completely missing the crucial data point that the amygdala needs to properly calibrate the threat.
Exactly. And the profound translational takeaway from the lesion studies is that if you explicitly verbally instruct an amygdala lesion patient to look at the eyes, if you force their visual saccade system to fixate on the eyes, you can temporarily reverse their deficit in fear recognition.
Which gives us a massive hint about potential behavioral therapies, which I definitely want to bring up at the very end of our discussion. But first, let's move to our final conceptual section: integration and clinical reasoning. We need to tie these network abnormalities to genetics, medications, and neurodevelopment. Let's start with the pharmacology. We have all these broken phone lines and short circuits. Do our antipsychotic medications actually fix the networks?
The data from pharmacologic fMRI is currently limited and complex, largely due to cross-sectional study designs and a severe lack of double-blind longitudinal imaging studies. However, the early evidence gives us some intriguing clues. When patients are switched from first-generation typical antipsychotics like haloperidol to second-generation atypicals like risperidone or olanzapine, researchers have observed some functional normalization of brain activity.
What does that normalization look like on the scan?
For example, during an end-back working memory task, patients who were switched to second-generation agents showed increased, more normal bold activity in the dorsolateral prefrontal cortex and parietal cortex compared to their baseline on older meds. We also see improved functional activation in circuits related to motor control and prepulse inhibition, which is another sensory gating mechanism.
But the textbook warns us to take these pharmacologic fMRI findings with a massive grain of salt. Why the caution?
Because, as we established earlier, bold is a relative measure. Without absolute baseline perfusion controls like ASL in these specific longitudinal studies, it's very difficult to definitively say the drug's mechanism of action directly repaired the neural circuit.
I see.
The increased prefrontal activity might just reflect general clinical stabilization. If the patient is less psychotic and less anxious on the new medication, they might just be trying harder on the memory task, which looks like normalization on the scan. We also have to worry about practice effects. They've taken the memory test multiple times now.
So the therapeutic mechanism remains a bit opaque. But what's really fascinating in the text is what happens when you use pharmacologic fMRI, not to look at treatment, but as a probe to uncover hidden genetic vulnerabilities. Tell us about the alprazolam study in unaffected relatives.
This is a beautiful, elegant use of pharmacologic probes. Schizophrenia has a strong genetic component, but many relatives carry the risk genes without ever developing the disease. Researchers wanted to see if those risk genes alter brain networks, even in healthy relatives. They took first-degree relatives who do not have schizophrenia and gave them alprazolam, a benzodiazepine.
Benzodiazepines act by enhancing GABA, the primary inhibitory neurotransmitter in the brain.
Yes. Now, during an emotion identification task in the scanner, these unaffected family members showed an abnormally strong, massive reduction in amygdala and hippocampal activation compared to healthy control subjects who were given the exact same dose of the drug.
Wait, they had a hyper-response to the GABAergic drug?
They did. It hints strongly that there are underlying, unmasked GABAergic abnormalities present in the family line. We know from postmortem studies that patients with schizophrenia have deficits in parvalbumin-positive GABA interneurons, which are crucial for synchronizing network activity and inhibiting runaway excitation.
So the genetic vulnerability, the GABA deficit, is there in the relatives, lying dormant and compensated for. But the pharmacologic challenge of the alprazolam disrupts that compensation and brings the hidden trait to light on the fMRI scan.
Exactly. It reveals the endophenotype, and that is a perfect bridge to genetics. The sources discuss the massive effort to incorporate genomics into functional imaging genetics, but they offer a stern warning about statistical power.
Right. They note that the effect size of any single genetic variant on an fMRI scan is incredibly tiny.
Absolutely. Schizophrenia has a highly complex polygenic architecture. It is not Huntington's disease. It's not one single dominant gene. It's thousands of tiny variations, single nucleotide polymorphisms, or SNPs, each contributing a microscopic fraction of risk.
But early researchers tried to link specific candidate genes to specific fMRI abnormalities. The textbook mentions the COMT Val158 MET substitution. Let's unpack the mechanism there.
COMT stands for catechol-on-methyl transferase. It is an enzyme that degrades and clears dopamine from the synapse, particularly in the prefrontal cortex, where dopamine transporters are scarce. The gene has a common variant. You can have the valin or val allele or the methionine ment allele. The val allele produces a highly active enzyme which clears dopamine very quickly, leading to lower dopamine levels in the prefrontal cortex.
And the met allele.
The met allele produces a sluggish enzyme, leaving more dopamine in the synapse.
And dopamine is crucial for the prefrontal cortex to hold information in working memory. So early on, researchers hypothesized that your COMT genotype would dictate how your prefrontal cortex lit up on an fMRI during a memory test.
And early small-scale studies seem to confirm this. They suggested individuals homozygous for the high-activity val allele had diminished inefficient prefrontal activation during working memory tasks compared to MET carriers. It was a beautiful story linking a molecule, a brain network, and a cognitive trait. But science requires replication. A recent massive meta-analysis combining data from thousands of scans found no reliable, statistically significant evidence for the impact of this specific COMT genotype on brain activation during working memory.
The signal just disappeared in the noise of a larger sample.
It highlights the extreme danger of small sample sizes and publication bias in imaging genetics. On the other hand, looking at polygenic risk scores or haplotypes in genes like GRM3, which encodes a metabotropic glutamate receptor involved in modulating synaptic transmission, has shown more robust correlations with reduced prefrontal and hippocampal activation during verbal memory.
Glutamate being the primary excitatory neurotransmitter, which ties into the NMDA receptor hypofunction hypothesis of schizophrenia.
Yes. But the overarching takeaway from the text is that to find these tiny genetic signals in the immense noise of a BOLD scan, you cannot rely on a single lab scanning 40 patients. You need massive international multi-site consortia pooling data from tens of thousands of individuals to achieve true statistical power.
Let's pull all this together by looking at the developmental timeline. Medlock Holmes has mapped the broken circuits in adults, the frontotemporal disconnect, the hyperactive amygdala. But the ultimate clinical question is, how early do these network issues appear? Are they a consequence of years of psychosis, or are they there before the first psychotic break?
This is where the vanguard of the field is currently focused, examining clinical high-risk or prodromal youth. These are young people, often adolescents, who are seeking help for attenuated subthreshold psychotic symptoms, fleeting perceptual disturbances or odd beliefs, but who have not yet developed full-blown schizophrenia.
They are in the crucial window of vulnerability. What does functional neuroimaging reveal about their networks?
It reveals that the network abnormalities are already present. Even before the full onset of the disease, prodromal youth show the exact same functional patterns we've been discussing in chronic adults.
So the architecture of the illness is laid down early.
Yes. They show decreased prefrontal activation during working memory tasks. They show increased, exaggerated amygdala responses to threat-related facial expressions. And when you apply graph theory to their resting state functional connectivity, they show multifocal disconnectivity. Specifically, they exhibit hyperconnectivity within the default mode network and diminished weakened coupling between the frontal executive networks and the default mode.
The endophenotype, the biological signature of the vulnerability, is established long before the first hallucination crystallizes.
Exactly. The underlying neurodevelopmental pathology is present throughout the central nervous system early in life. But this raises a profound mechanistic question for the listener. If the entire brain carries these genetic risk alleles and the entire brain is exposed to whatever early environmental hits occurred, why are some specific circuits so severely affected, like working memory in the DLPFC and social cognition in the amygdala, while other circuits like basic motor control or primary visual processing in the occipital lobe remain relatively spared?
Why does the telephone exchange only misroute certain calls?
It comes down to one fundamental neurobiological concept: plasticity. The brain regions that are most deeply vulnerable and functionally altered in schizophrenia, the hippocampus, the prefrontal cortex, the extended limbic system, are precisely the regions that mature very late in human development, undergoing massive synaptic pruning and myelination well into adolescence and early adulthood.
They remain under construction much longer.
Yes. They are highly dynamic, and they must maintain a massive degree of neuroplasticity because their job is to constantly adapt to complex, ever-changing environmental demands and highly nuanced social learning.
Because they are highly plastic. Because they are so moldable and adaptable for so long, they are inherently fragile.
That is the crux of it. This prolonged state of plasticity makes them incredibly vulnerable to neurodevelopmental aberrations. If there is a subtle genetic vulnerability involving glutamate or GABA, or an environmental hit like maternal infection or adolescent cannabis use, it is these late maturing, highly plastic regions that bear the brunt of the damage as the brain attempts to wire itself during adolescence. Functions mediated by older, less plastic, early maturing neural systems like basic motor or sensory processing don't require that same prolonged modifiability, so they lock into place early and escape relatively unscathed. Schizophrenia is fundamentally a disorder of neuronal plasticity gone awry during the brain's final, most complex stages of maturation.
That is a brilliant synthesis of the entire chapter. Okay, let's step back and summarize the journey Medlock Holmes has taken us on today in the Hall of Living Minds. We started by moving past the frozen architecture of structural scans and into the living, pulsing networks revealed by fMRI.
Right, measuring the physiology.
We learned that we don't measure neurons directly. We track the catering trucks of oxygenated blood using the paramagnetic properties of deoxyhemoglobin and bold, and we establish absolute baselines using ASL. We discovered that cognitive deficits aren't just dead zones, they are broken conversations. Specifically, we see reduced connectivity between the dorsolateral prefrontal cortex and the temporal lobes. The manager and the worker cut off from each other, resulting in ineffective compensatory calls to the ventrolateral cortex.
The misrouting of resources.
And perhaps most surprisingly, we uncovered the irony of the amygdala. Flat affect isn't a lack of emotion. It's a deafening internal alarm system going off so intensely to ambiguous threats that it short-circuits top-down cortical processing, leading to both misidentification of fear and a compensatory protective emotional shutdown externally.
An overload, not a void.
We trace these vulnerabilities from complex polygenic architectures through GABAergic deficits unmasked by alprazolam, all the way to the resting state disconnectivity in prodromal youth. And we finally realized that the late maturing, highly plastic regions of the brain are the ones most susceptible to this developmental derailment.
It is a remarkable testament to how functional imaging has transformed our clinical conceptualization from a static, localized lesion model to a dynamic, distributed network model.
And before we get to our learning reinforcement, I want to leave you with a final thought to mull over. Building on what we discussed regarding the amygdala, the magnocellular visual pathway, and eye contact. We know that the failure to process fear in schizophrenia is heavily linked to patients actively avoiding the eye region of faces. And we know from classical lesion studies that simply instructing a patient to look directly at the eyes can temporarily reverse their deficit in threat recognition. So could the future of schizophrenia treatment look less like a pill and more like targeted behavioral visual saccade training? Imagine a therapy protocol using real-time fMRI as biofeedback, teaching patients to consciously redirect their gaze to the eyes of avatars, and watching on a screen as that simple mechanical movement begins to rewire and calm their hyperactive amygdala. Imagine rewiring the mind's threat detection system, simply by teaching the eyes where to look.
That is the ultimate promise of translational neuroscience, moving from observation to targeted, network-level intervention. And now, to ensure these concepts are deeply encoded in your own frontotemporal networks, let's move to our single best answer questions.
These questions are designed to test your conceptual understanding and clinical reasoning, not just word memorization. Grab a mental notepad. Question 1. We spent a lot of time on the physics of our instruments. Which of the following best describes the physiological basis of the blood oxygenation level dependent, or BOLD, signal in fMRI? A relative increase in deoxyhemoglobin leading to signal enhancement. B, an increase in regional blood flow that exceeds regional oxygen consumption, resulting in a local decrease in deoxyhemoglobin. C, direct measurement of electrical action potentials via magnetic induction. Or D, absolute quantification of cerebral blood flow using magnetic tagging of arterial water.
Let's think through this. We can eliminate C straight away. BOLD is a proxy, not a direct electrical measure. Option D describes arterial spin labeling, or ASL, not BOLD. That leaves A and B. Remember the physics. Deoxyhemoglobin is paramagnetic and decreases the signal. Therefore, option A is incorrect. The correct answer is B. When a brain area activates, neurovascular coupling causes the vascular system to overcompensate, flushing the area with highly oxygenated blood. This massive relative drop in deoxyhemoglobin is what actually increases the signal intensity we read on the scan.
Excellent. Question 2. This focuses on experimental methodology in clinical confounds. When attempting to study the neural substrates of a task in which patients with schizophrenia are historically impaired, such as facial effect identification, why might an investigator choose an event-related fMRI design over traditional block design. A. Event-related designs provide a higher signal-to-noise ratio for robust network mapping. B. Event-related designs are less susceptible to head motion artifacts. C. Event-related designs allow the investigator to separate neural activation associated with correct responses from activation associated with incorrect responses. Or D. Event-related designs require significantly less time in the scanner, reducing claustrophobia.
Well, while D might be nice for the patient, it's not the primary scientific reason, and A actually describes the strength of a block design, which integrates signal over time. The correct answer is C. Block designs aggregate everything, meaning you are hopelessly mixing the neurobiological signal of the core illness with the state artifact of failing the test, including anxiety and frustration. Event-related designs time-lock to individual stimuli, allowing researchers to isolate accurate processing. This levels the playing field to see if the circuitry remains fundamentally altered even when the patient successfully completes the task.
Question 3. Let's look at the broken conversations of cognition. In studies of verbal episodic memory encoding in schizophrenia, functional connectivity analyses using time series correlations typically reveal which of the following patterns compared to healthy controls. A. Increased connectivity between the dorsolateral prefrontal cortex, or DLPFC, and temporal regions. B. Reduced connectivity between the DLPFC and temporal regions, and increased connectivity between the ventrolateral prefrontal cortex, or VLPFC, and temporal regions. C. Global hyperconnectivity across all frontal and temporal nodes. Or D. Isolated hypoactivation of the hippocampus with intact frontal connectivity.
The clinical data points us directly to B. The reduced DLPFC temporal connection is the core broken conversation, the cut phone line that correlates directly with episodic encoding deficits. The simultaneous increased VLPFC connection represents the ineffective compensatory effort. The brain is trying to dial the backup manager, but this alternate routing fails to solve the functional problem, resulting in poor memory recall.
Question 4. This touches on the paradox we explored. During event-related fMRI studies evaluating facial emotion identification, how does the amygdala response of patients with schizophrenia typically differ from healthy controls when presented with threat-related expressions like fear and anger? A. Patients show a diminished amygdala activation, which correlates with flat affect. Patients show an exaggerated bottom-up amygdala activation, which is strongly associated with incorrect identification and more severe flat affect. C. Patients show normal amygdala activation, but profound visual cortex hypoactivation. Or D. Patients show amygdala activation only to neutral faces, not fearful faces.
If you rely purely on clinical intuition based on the outward appearance of the patient, you would naturally guess A. But the imaging data reveals the irony of the amygdala. The correct answer is B. Unlike healthy controls where a strong limbic response helps them correctly identify fear, patients experience a massive, exaggerated bottom-up interference. The alarm is so loud it short-circuits the top-down cortical categorization, leading to multiple-choice errors and correlating strongly with the compensatory negative symptom of flat affect.
And finally, question 5. Looking at the neurodevelopmental trajectory, resting state functional connectivity analyses in youths at clinical high risk, or prodromal for psychosis, generally demonstrate which of the following network abnormalities. A. Completely normal network topology until the first episode of psychosis. B. Isolated hyperconnectivity of the primary motor cortex. C. Hyperconnectivity among default mode regions and diminished coupling between frontal executive and default mode networks. Or D. Massive global reduction in all functional edges across the brain.
The correct answer here is C. The data clearly shows that network topology is not normal prior to the first break, which eliminates day. The multifocal disconnectivity observed in prodromal youth, specifically the failure to properly couple the internal default mode network with the external executive networks, perfectly matches the network abnormalities seen in adults with established chronic psychotic disorders. This proves that these network derangements act as early developmental endophenotypes linked to the aberrant plasticity of late-maturing brain regions.
And that brings us to the end of our time in the Hall of Living Minds. This was an intense conceptual journey. Be sure to read alongside Chapter 12.8 of Kaplan and Sadok to cement these models, look at the actual fMRI images provided in the text, and keep your minds curious until next time.